In last video, I told you about the DNA, the structure of different nucleoside and nucleotide. Today, we start from this, the structure of DNA. Right? In last video, already we discussed about the nitrogen base, about the structure of sugar, pentose sugar, both sugar, ribose and deoxyribose, and phosphoric acid. Right? Then after we discuss about the nucleosides, different nucleosides, and then different nucleotides. Right? What is the structure of different nucleotides? At that time, we understand that phosphoric acid, that is H3PO4, it is always binds with pentose sugar with its fifth carbon, right? And nitrogen base, it binds with pentose sugar through the first carbon, right? Nitrogen base, when it binds with the first carbon, it forms an glycosidic bond, and when phosphoric acid, it binds with the fifth carbon, it forms phosphoester bond, right? Now here, some other points are also how a nucleotide chain is formed. We can say how a strand, DNA single strand is formed, either the DNA or RNA, how a strand is formed. Today first I tell you how a strand is formed, right? Then how the two strands bind with each other or connect with each other through hydrogen bond, we will also discuss about this time, right? Now we are going to discuss about, then after we start this. Okay, so first here, look here how a nucleotide strand of backbone of DNA is formed. How a nucleotide structure attached with another nucleotide, or we can say how a backbone of DNA or RNA, or we can say how a strand is formed. You can see here, suppose that I am I am going to draw a nucleotide that is called adenylic acid, right? We know this is the base structure of purines, because adenine is a purine, right? That is double cycle structure. Here, adenine, it contains an H2 group, right? Now this is the position of nitrogen. Already I discussed you the last, right? This is the position of nitrogen. When we can, all these other, this is the carbon, here also carbon, and this side carbon. Look here, 1, 2, 3. Now we need one more, than 1, 2, 3, 4 valence is completed, nitrogen 1, 2, 3 completed. This is the carbon because it is adenine, right? So it contains here double bond, right? 1, 2, 3. No, it contains here CH, right? It contains here N group, right? This one is N. Here it is double bond O. 1, 2, 3, 4. Nitrogen 1, 2, 3. 1, 2, 3. 1, 2, 3. Nitrogen 1, 2, 3. And C H 1, 2, 3, and 4. This nitrogen H is there. Already we discussed this structure, right? Now here, when a pentose sugar is binds with adenine, what happens here? This one is pentose sugar. This is the first carbon, second carbon, third carbon, fourth carbon, and this one is the outermost fifth carbon, right? This first carbon, it's bind with, look here, this H2O is removed. H2O is removed, and here which bond is formed? Here N glycosidic bond is formed, right? N glycosidic bond is formed. Now look here, okay, this is the second carbon, third carbon, right? This third carbon, it contains here OH group, right? This side H is there. So this is a single nucleoside, and or we can say this is the structure of adenosine. When a phosphoric acid is attached with adenosine, it becomes nucleotide, that is adenylic acid. So this is the fifth carbon. The fifth carbon, this one is the fifth carbon. Now this fifth carbon, this is the fifth carbon, this is the first carbon we know, right? Fifth carbon. Now this fifth carbon attached with phosphoric acid. Suppose that I draw here this phosphoric acid H3PO4 and OH. Now you can see here CH2, this H2O is removed from here, right? When H2O is removed here, then what happens here? Phosphoester bond is formed. Phosphoester bond is formed. Which phosphoester bond? Because fifth carbon is attached here, we can say 5' phosphoester. Now you understand here, this side it is called 5'. 5' means it is at 5' terminal and phosphoester bond or phosphoric acid, right? Now look here, the third carbon. This one is our third carbon. Now this is the structure of a nucleotide, and this nucleotide structure, what is the name of this nucleotide? So we can say here, this one is AMP. AMP means what? Adenosine 5' monophosphate, right? Or it is the another name of this one is adenylate acid, right? Because it is made up of adenine. This one is ribose sugar because it contains, look here, the second carbon, if second carbon contains OH group, then it is ribose sugar. If second carbon it contains only H, then it is deoxyribose sugar, right? Okay, now this is the nucleoside. How does it binds with another nucleotide, or we can say dinucleotide? How a dinucleotide is formed? So here second nucleotide, here this one the second nucleotide we can say here. Okay, I move this one some above direction, then we can easily prepare this one, right? Because we need some more space, so I scroll this one. Now look here, this one is another. Another means what? It may be, because it is a single strand, so it is nucleotides are present in randomly. No sequence is there, right? So here what happens? Another, suppose that I draw here a thymine structure, right? This one is our thymine or cytosine, any nucleotide we can look here, right? Look, this is the third carbon. This third carbon binds with another phosphoric acid. This one is phosphoric acid. Double bond O, it is OH, OH and OH, right? Now can you see here? Here H and OH. One H2O is removed, right? Because it is also third carbon, oxygen, phosphorus is there. It is the another phosphoester bond is there, because two phosphoester bond is present in a single, right, pentose sugar. So it is called as diphosphoester bond or phosphodiester bond we can see here, right? Now this is the phosphorus, it attached with the another pentose sugar. I draw here the pentose sugar, right? You understand, this is the CH2OH, this is the fifth carbon, this is the fifth carbon, this one is attached with this fifth carbon, one H2O is released, here another phosphoester bond is formed, ester bond is formed, right? You understand this, how a structure is formed? Because this is the first carbon, this is the fifth carbon, now 4, 3, 2, 1, this is the first carbon, OH is there, right? And we know this first carbon is bound with another nitrogen. Another nitrogen base is here, it is our, suppose that this one is our pyrimidine. Pyrimidine means this first nitrogen binds with first carbon, right? So this first carbon means here, this H2O is removed. This H2O is removed. And when H2O is removed, what happens? Here, N, because this side N, this side carbon, it is formed N-glycosidic bond is formed. N-glycosidic bond is formed, right? here this one it is carbon, this one is nitrogen, right? Now it is depend on, suppose that this one is a you say sir it is thymine or adenine. So suppose that I made it thymine, it is thymine or cytosine, maybe cytosine. Thymine means what here? If this carbon, it is not a first, this is first 1, 2, 3, 4. So this fourth carbon, if this fourth carbon or 1, 2, 3, 4, it is fourth, right? The fourth carbon, if it is binds with double bond O, then this one is thymine, or if it is bind with NH2, then it is cytosine, right? So it is depend upon the atom which is bind here. Suppose that I form it is a thymine, that means it is double bond O, right? Here NH2 1, 2, 3, 4, NH, this side double bond O. Now here you understand this N-glycosidic bond is formed here, right? So this how A form. Look here, in this case, this single phosphoric acid, right, on both sides it binds with two pentose sugars. This phosphate one side to it is the this phosphoric acid, it belongs to this one, right? Suppose that I write here, this is the first adenosine A, and this one is because I draw here double bond O. This one is thymine, right? This side thymine means what? It is CS, right? This side C is there, double bond. Okay, I already tell you this one, right? Now it is thymidalic acid. Thymidalic acid, or it is also called as TMP. Thymidalic acid, because it is a thymidalic acid, you understand this one is not a RNA, it is a DNA strand, because thymine is absent in RNA. You understand, right? So it is also called as TMP. TMP means what? Thymidine 5-monophosphate, right? Thymine. Deoxythymidine, we should write this one deoxythymidine. Why? Because it is a DNA. Because thymine is present in DNA. That means what? This pentose sugar, it contains only H in the second carbon, right? And here it is OH and H. So you understand here, phosphoric acid, it attach, okay, with its nucleoside, that is through fifth carbon, and toward the other nucleotide, it attach with the third carbon of the pentose sugar. So 3 and 5, that's 3 and 5, that 3 and 5, it represents the direction, right? Now you see in this case, right, this is the O, right? What happens? This side 5 dash is present above side, and this one 5 dash it is the upper portion, 3 dash it is the lower portion. What is the direction of this strand? We can say here 5 dash, we should write here 3 dash. Upper side is 5 dash, lower side is 3 dash, right? Then this one is 5 dash 3 dash direction, right? And this one, this structure, it shows how a strand is formed or how a backbone is formed. Look here, these nitrogen base are not attached with each other here. Only phosphate, we can say sugar phosphate backbone, because sugar attached with phosphate, then a phosphate attached with sugar, another phosphate attached, then another sugar, another phosphate, another sugar. Now A means we can say alternate term is given, sugar phosphate, sugar phosphate, sugar phosphate. Now this backbone is called sugar phosphate. Sugar means it is pentose sugar, either ribose or deoxyribose, sugar phosphate back. Right? You understand how a dinucleotide is formed, because here phosphoester, one phosphoester bond is there, another phosphoester bond is there. So we can say here it is a phosphodiester bond is formed, right? Okay, you understand? Now second and last thing what I want to tell you, that how the hydrogen bonds are formed between two strands of DNA. It may be formed between means if a folding occurs in RNA, in foldings are here also hydrogen bonds are formed, right? As for example, in transfer RNA, you understand here, right? Transfer RNA, when it folds, they form the hydrogen bond in between them, right? Except only last point means where means abnormal nucleotides are there, they cannot bind with, right? Otherwise, other foldings means it is okay. Now here, look how a di or trihydrogen bonds are formed between nucleotides. Adenine and guanine, in between adenine and guanine, they are purines, and cytosine, thymine. In case of DNA, cytosine and thymine, they are pyrimids, right? Chergaff said purines are always equal to pyrimidine, or we can say also pyrimids are always equal to purine, right? That means A is always equal to P and C is always equal to G, or we can say ratio is equal to 1. That is called equilibrium law of Chergaff, okay? And it is only for DNA. Look here, suppose that it is a adenylate acid. I draw here only the specific structures, not a detailed structure, but which are responsible for that is NH2. We can write, we should write here, right? Because here NH2 is there is carbon 1, 2, 3. We need one more, that is N, right? Okay, this side N is there. So we need not go to discuss very detailed structure, only how a tri and dihydrogen bonds are formed here, right? So this one is adenine. You understand how it is adenine, because this first carbon, this one is the first carbon, right? And this side it is H. This side H. See, this is the first carbon, because how? Because first carbon contains amino group, that means it is adenine, right? Okay. Now here, second, adenine always binds with this side. It is pentose sugar. This side is pentose sugar. We know. First carbon means what? OH group, and a water molecule is released here. I already tell you, right? This one is pentose sugar, and that side it is. This is the fifth carbon, CH2OH, and I tell you, phosphoric acid is attached with the fifth carbon, this carbon. So this is the phosphoric acid, P double bond O, OH, OH, and OH. You understand here? This H2O is released, and here phosphoester bond is formed. So this is a nucleotides, simple structure. Look here, our main point is here. Adenine, this one is adenine because it contains amino group. So I write here adenylate acid. Correct? You understand what is the structure? This is the structure of adenylate acid, right? Now this adenylate acid, it binds with thymidylate acid, because A always binds with T through 2 or 3 only 2 hydrogen bonds, right? So this is our that side C, it, because this NOH, so this C, it contains double bond O, right? This side it is N. So 1, 2, 3, 4 completed, this N contains H, right? This side is double bond O. This side N, right? CH3. This one is CH3, and this side H. Right? This you understand? That one is always also bind with pentose sugar. We can say, right? I draw here pentose sugar. And now this pentose sugar is binds with H3PO4. Here H3PO4. We can say it is another nucleotide. I did not write like so huge structure. Now look here, how a bond is formed. One hydrogen bond forms with this O. Another hydrogen bond forms between N and this H. So these two bonds, these are hydrogen bonds. These two, right? This one and this one. These two are hydrogen bond. So in case of adenylic acid, and this one is thymidyllic acid. How many bonds are formed? How many hydrogen bonds are formed? 2 hydrogen bonds are formed. One in between O and this ketone group and H, right? Another one is in between N and H, right? Now here, second, here I am going to draw The another means what nucleotide? It is, we have only 4 nucleotides, that is cytidalic acid and guanine. See here how it means a triple bond is formed. This is our guanilic acid, and this side I draw, it is our cytidalic acid, guanilic and cytidalic acid, right? Look here, guanilic acid, it contains double bond O. We know very well, double bond O is there, right? Double bond O. This side, this one is the N. This side, it is N, N, and these are the position of N, right? Because look here, 1, 2, 3, 4, all the carbon valence is completed. Here 1, 2, we need one more, that is NH. Now it is completed. This side, because it is guanilic acid contains, this is CNH2, right? NH2, NH2, this like NH2, right? Now here, this is the NH, here pentose sugar, 5th carbon bind with pentose sugar, 5th carbon CH2OH is there, right? No, this is the first carbon, not 5th carbon. This one is the first carbon, C and N. It is glycosidic bond, right? And this side, it is the 5th carbon, CH2OH. It bind with phosphoric acid, that is POOH, and this side, okay, I remove this one, right? You understand? Already CHO, this one, right? Same structure, both are same structure. It is the 5th carbon, right? Now look here how it binds with this side, because this one is our guanilic acid. Guanilic acid, right? Because here it contains ketone, this one O. Now this side, it is, we know G is always bound with C with triple hydrogen bond. So C means it contains, here it contains NH2, right? This NH2, this carbon, this is the nitrogen, right? Because it 1, 2, 3 and 4. So this completed nitrogen is also completed. This side is carbon, here double bond O is there, this side nitrogen, and this is binds with another pentose sugar, because it is another nucleotide, right? And this is the 5th carbon, CH2O, and it is binds with P double bond O, OH and OH. That is another nucleotide, same as to this one, right? Here 5th carbon, this is CH2H, right? CH2O. Now look here, first carbon, this binding, first carbon, second, third, fourth, this side O, this side O. We should write, otherwise you get confused. Now there is no confusion, I think, right? This is the O. N. Okay. This side H. Now look here how a triple bond is formed. This one hydrogen bond formed between this oxygen and hydrogen. Second hydrogen bond is formed between this H and this N, right? Not this H, this H and N, right? Nitrogen and hydrogen. Third hydrogen bond is formed between this one, right? This one. So one, this is the one first hydrogen bond, this is the second hydrogen bond, and this is the third hydrogen bond. So 1, 2 and 3. Triple hydrogen bond is formed between guanilic acid, and this one is cytidalic acid. You understand how a strand is formed, how a hydrogen bond is formed. Adenine bind with thymine with double hydrogen bond, and guanine bind with cytosine through triple hydrogen bond, right? Now you understand, right? Okay, now here, so this is the two, we can say two, these are called base pair. base pair between A double bond T and this side it is G triple bond C. Okay. Now here, when a sugar phosphate bond, how it is formed? You can see here, this is the phosphoric acid, it is the fifth carbon. Now here it is the first carbon here, second carbon is this one, and this one is the third carbon. Third carbon means it contains OH group. Now you can understand here, this OH and H, when it removes, so what happens here? A phosphoester bond is formed here. Phosphoester bond is formed, right? And a sugar phosphate bond is formed. You can understand here. This one is the one strand. Another side also same thing is there. This is the first carbon. We can say first carbon. This one is N-glycosidic bond is there, right? And this side, this is the fifth carbon. This is the fifth carbon. Here it is fifth carbon. Now fourth carbon, third carbon, second carbon is one, and this one is the third carbon. This third carbon, I remove this acid, then you understand, right? This is the third carbon. Third carbon, it contains OH group. Now here, this OH and this OH, what happens here? This H and this OH, right? One H2O molecule is released, and another here phosphoester bond is formed. This is the another sugar phosphate backbone is formed. Here you understand. Now it is a dinucleotide, or we can say small strand. Okay. Now this process continues, and a large molecule, a large polymer of polynucleotides, we can say polynucleotides, a large polymer of nucleotide is formed, right? And a double helical structure is formed. Okay. In case of, when we see what is the model of bDNA, because Watson and Crick, they propose the bDNA model. Here you see this model of DNA. Then Watson and Crick, he gives a simple model, double helical structure, right? This like structure. One, these are the nitrogen base, we can say. Okay. Randomly, I write here A, G, C, E, A, E, E, C, G. But we have to calculate 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Right? Then A. So from this means half to another half. Right? It contains 10 base pair according to Watson and Crick in bDNA. This is the structure of bDNA. Right? So 10 base pair in one term. In one term. Ten base pairs are present in one term, right? And the length of this one term is 34 angstrom, or we can say it is 3.4 nanometer. Because in NCERT, nanometer is given. It is the nanometer means what? A nanometer is equal to 10 power 9 meters. And when we consider the angstrom, a angstrom, it is equal to 10 power minus 10 meters. Minus 10 meters. It is the 10, right? Here, 10 power minus 10 meters. That's why here 334 angstrom or 3.4 nanometer. It is the length of one work or one term, right? Because 10 base pairs are present. So between these two base pair, what is the length of this two base pair between two base pair? Simply when we divide this 34 angstrom with 10, then what happens? We get 3.4 angstrom. The distance between two nucleotides is 3.4 angstrom, or we can say 0.34 nanometer. Okay. Another thing what he give us, it is the distance between two strands. These two strands, right? This distance. So this distance is, he said, it is 20 angstrom, or when it is in nanometer, we can say it is 2 nanometer. Right? Complementarity is there. If we have one strand sequence is there, we can understand what is the sequence of another DNA, because they are complement to each other. Right? Adenine is this side, adenine is there, that means here it is thymine. Guanine, that means here it is cytosine. Cytosine, that means here guanine, thymine, adenine, adenine, thymine. Right? Thymine, adenine, cytosine, guanine, cytosine, adenine, thymine, and so on. Right? You understand this one. Adenine, already I tell you, binds with thymine through double bond, and guanine always binds with cytosine through triple bond. Adenine is always equal to thymine, and guanine is always equal to cytosine. That means if I write here A upon T is equal to 1, that is correct, or C upon G, C is equal to 1, that is also correct. Right? Or do you understand this one? You understand? This is the model given by Watson and Crick. So they get Nobel Prize about this DNA structure, and this is a DDN. One thing, another, what he said polarity. When we see the polarity of these two strands, they are just opposite to each other, or we can say they are anti-parallel. Anti-parallel means what? If one strand is 3 dash 5 dash, this one is one strand, it is if 3 dash 5 dash, that means another strand is 5 dash and 3 dash. Okay, you can see here, suppose that I draw another line is there in another color, you can easily right, so you can easily understand that one, which one is the strand. So two strand means two different antipolar. This means what? If this one is arrow is this side, that means this arrow is from that side. Just opposite direction. Okay. So this is the model, one means DNA model of Watson and Crick. Now come back to the NCERT. What is given in NCERT, because we have to follow the NCERT. So here I think already we read this, the DNA. The DNA is a long polymer of deoxyribonucleotide. Already we know, understand how a DNA polymer is formed, right? Deoxyribonucleotide. Why it is called deoxyribo? Because here ribose sugar is, here the pentose sugar is deoxyribose sugar. And already we know what is the difference between deoxyribose and ribose. In deoxyribose, second carbon has no oxygen. It lacks oxygen. But in ribose sugar, it contains OH group. Means oxygen is present here. Right? The length of DNA is usually defined as the number of nucleotides or a pair of nucleotides referred to as base pair here present in it. Number of nucleotide means if one nucleotide add, that means 3.4 angstrom. This means length is add. Right? This also is characteristics of any organisms, because different organisms have different number of base pairs. For example, of a bacteriophage known as I-174 has 5386 nucleotides. Confirmed number is there. Bacteriophage lambda has 48502 base pairs. Right? Escherichia coli has 4.6 into 10 power 6 base pairs. And human haploid contents. Haploid means in gametes. Right? Haploid content of human DNA is 3.3 into 10 power 9 base pair. That means what? If it is diploid content of human, when we consider the diploid content of human, we have to multiply with 2. Double number of portion. When we multiply with 2, that means it becomes 3.3 becomes 6.6 into 10 power 9 base pair. Okay. Let us discuss the structure of such a long polymer. The structure of a polynucleotide. Let us recapitulate the chemical structure of a polynucleotide chain, DNA or RNA. A nucleotide has three components. Already in first lecture, I tell you, a nitrogen base, right? A pentose sugar, ribose in case of RNA, and deoxyribose for DNA, and a phosphate group, H3PO4, right? There are two types of nitrogen base. One is called purine, and purine contains adenine and guanine. And pyrimidines. Pyrimidine is single cyclic structure. They contain three types of nitrogen base, that is cytosine, uracil, and thymine. In case of RNA, uracil is there. In case of DNA, thymine. Okay, that is cytosine, uracil, and thymine. Cytosine is common for both DNA and RNA. Cytosine is present in both, right? But thymine is present in DNA only. That means thymine is present in the form of deoxythymidyllic acid only, right? Uracil is present in RNA at the place of thymine. That means in RNA, it is only uracil is there. We can say uridylic acid is there. Here deoxy is not there, right? Because it is present only in RNA, ribosome. A nitrogenous base is linked to OH group of first carbon pentose sugar. Already we draw the structure and tell many times. I tell you, first carbon of pentose sugar binds with the which one? Nitrogen base OH group. If it is a purine, means ninth nitrogen binds with the first carbon of pentose sugar. If it is pyrimidine, then first carbon of pentose sugar binds with the first carbon of nitrogen base. Correct? Sugar through N-glycosidic linkage to form a nucleoside. When nucleoside means what? One nitrogen base. When nitrogen base and nitrogen base and pentose sugar bind with each other, then this structure is called nucleoside. And when nucleoside binds with phosphoric acid, then this structure is called nucleotide. Right? We can say nucleotide means what? Nitrogen base, pentose sugar plus phosphoric acid. Right? Or it is also, we can say, nucleoside plus phosphoric acid is a nucleotide, because nucleoside means what? Nitrogen base and pentose sugar. Right? N-glycosidic linkage to form a nucleoside such as adenosine or deoxyadenosine. Adenosine means when sugar is ribose sugar, we can say adenosine. It is present in RNA only. And when ribose sugar is deoxyribose, then it is called deoxyadenosine, which is present in DNA. Guanosine, RNA, or deoxyguanosine, DNA. Cytidine, RNA, deoxycytidine, DNA. And uridine, only in RNA. And deoxythymidine, only in DNA. Okay. When a phosphate group is linked to OH group of fifth carbon, look here, phosphoric acid or phosphate group, it binds with fifth carbon of pentose sugar. Already we know this one, right? And I will tell you this one, right? Of a nucleoside through a phosphoester linkage POC, right? Then a corresponding nucleotide or deoxynucleotide is formed depending upon the type of sugar, pentose sugar, right? So nucleotide or deoxynucleotide is formed. If it is ribose sugar, nucleotide is formed. If it is deoxyribose sugar, then it is deoxyribonucleotide is formed. Okay. Two nucleotides are linked together. Now, the two different nucleotides, that means suppose that it is adenyllic and the other one is thymidyllic or cytidyllic. So they linked through 3-5- phosphodiester linkage, right? Because a single sugar, pentose sugar, its fifth carbon has its own phosphoric acid and its third carbon binds with another nucleotide's phosphoric acid. The third and fifth carbon both are bind with phosphoric acid, so it is called 3-5- phosphodiester linkage to form a dinucleotide. Right? Here we can say a sugar phosphate bond, a sugar phosphate backbone is formed. Right? This 3-5- phosphodiester linkage, it forms the sugar phosphate backbone, or it is responsible for the formation of strand for RNA as well as DNA. More nucleotides can be joined in such a manner to form a polynucleotide. A polymer thus formed has at one end a free, this side you can see here, phosphate moiety, 5' phosphate, means this is phosphoric acid. right? And this is our fifth. This one is, it is the fifth carbon. This is a single strand, right? Now, can you tell me it is a DNA strand or it is a RNA strand? You have a strand. You can see the polynucleotide chain. Now, can you tell me it is a RNA strand or DNA strand? This clue is given because we know here thymine is given. This thymine, so it indicates that this DNA belongs to DNA, not RNA, right? Due to the presence of thymine, it is here. If it is here, uracil is there, we can say here uridine, right? Uridylic acid is there. Then we can say it is RNA. Okay, so this is a DNA strand. DNA polynucleotide chain is there. This side, you understand, this is the fifth carbon. This one is the fifth carbon of pentose sugar. This is the pentose sugar, right? You can see here first, second, third, fourth, and this is the fifth carbon. And fifth carbon bind with phosphoric acid, right? It is the called 5-phosphate moiety. This side, another side, and you can see here this one is a one nucleotide, another one is second nucleotide. They are here where the phosphoester bond is formed. Here one phosphoester bond is formed here. Phosphoester bond is formed. Another phosphoester bond is formed here. Phosphoester bond is formed. So this side it is third carbon. This side it is the fifth carbon. You understand this is the fourth. So third, fifth, we can say what is the direction of this nucleotide? It is 5 dash. You can see the 5 dash, and this side it is 3 dash. Look here. When you come to here, this is the fifth carbon and this is the third carbon. Third carbon has OH group. Last, because it has OH group, it has no phosphoric acid here. So because your last means 3 dash hydroxyl moiety is there. You understand? You understand? This is the itself. Now this phosphoric, right? It belongs to this thymine dilic acid. This phosphoric guanylic acid. This phosphoric satidilic acid. And this is the another phosphoric acid. It binds with the third carbon of pentose sugar, right? So do you understand this one? What is structure? Moiety. 3 dash means hydroxyl group is there. 5 dash means phosphoric acid is attached there. So phosphate moiety is there. Okay. This one is adenine. How you understand this one is adenine? Because we know here double cyclic structure is there, but here single hexagonal cycle, this single cycle is there. So we understand this one is purine. This one is purine made up of 2 cycles, and this one is pyrimidine. Right? Okay. Phosphate moiety at 5 dash end of sugar. You understand? Which is referred as 5 dash end of a polynucleotide chain. This, which is referred 5 dash end of a polynucleotide chain. Right? And similarly, at the other end of polymer, the sugar has a free OH. OH means hydroxyl group at third carbon. At third carbon, right? So third carbon, either it is a deoxyribose or ribose sugar. We know third carbon it contains OH group. Okay. Only second carbon difference is there. Second carbon means deoxyribose means what? Second carbon it lack oxygen, and ribose sugar means it has oxygen. OH group is there. Okay. So OH group which is referred as 3 dash end of the polynucleotide chain. The backbone of the polynucleotide chain is formed due to sugar phosphates. So it is called as sugar phosphate backbone. The nitrogenous base linked to sugar moiety project from the backbone. This is the backbone. Project means this one is nitrogen base, nitrogen base, nitrogen base. This is the nitrogen base. This side phosphorus, 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 phosphorus. This is our sugar. This one is phosphorus. This is the phosphorus. That's sugar, then phosphorus, then sugar, then phosphorus, then sugar. So this is the structure. This nitrogen toward the inside, right? In RNA, every nucleotide residue has an additional OH group present at second position of the ribose. Second position means second carbon. Many times I tell you here, second carbon contains OH group, that is given here. That's why it is unstable. This OH group makes it unstable. Okay. Also in RNA, the uracil is found at the place of thymine. Second is difference. Uracil is present in place of thymine, and this thymine is also called as 5-methyl uracil, because already when in the previous lecture I tell you, when I tell you the structure of uracil and thymine, at that time I tell you, right, in place of H group in thymine, it is CH3. CH3 means methyl group is present, and this methyl group is present in 5th carbon, so it is called as 5-methyl uracil. In uracil, CH3 group is absent, here only H is there, right? 5th carbon it contains only H, but in thymine 5th carbon it contains CH3. CH3 means what? Methyl, so it is called 3-methyl uracil also, right? It is another chemical name for thymine. Another chemical name of thymine is 5-methyl uracil. 5 indicates that this methyl group is attached with the 5th carbon of uracil. Okay, that is the difference. Uracil means only H group. 5th carbon contains H, I mean hydrogen. But thymine means what? 5th carbon of uracil, it contains methyl. That is called 5-methyl uracil, or it is another name, we give them thymine. Okay. DNA as an acidic substance. Acidic substances. What is acidic? Due to the presence of phosphoric acid, right? Present in the nucleus was first identified by Friedrich Miescher. Very important. In 1869, first Friedrich Miescher, he identified this DNA, but at that time DNA name is not given. He said them nuclein, right? This nuclein. Nuclein means what? In means what? Protein. He thinks it is a polymer, it may be protein. So it gives a protein which is present inside the nucleus. So it is called them, he called them nuclein, right? However, due to the technical limitation in the isolation of such a long polymer intact, the elucidation of the structure of DNA remained elusive for a very long period of the time, because we have at that time we have no more we have many technical limitation is there, right? We are not advanced in technology at that time, so the structure of DNA is not confirmed at that time. It was only in 1953 that James Watson and Francis Crick, long time is, you can understand, 1869 and then 1953, how much time it takes? 1869 Friedrich Miescher identified this nuclein, right? And near about to 1953, near about to 90 years after, we have no idea about the structure of DNA, right? In 1953, James Watson and Francis Crick, based on X-ray diffraction, you can see here, Watson and Francis Crick, based on X-ray diffraction data which is produced by Ulrich Wilkins and Rosalind Franklin, these two scientists, right? X-ray diffraction, who? Ulrich Wilkins and Rosalind Franklin, they produce this data, right? By using of this data, Watson and Crick in 1953, they proposed a very simple but famous double helical structure or double helix model for the structure of DNA. One of the hallmark of their proposition was based pairing between the two strands of polynucleotide chain, right? They first proposed this double helical structure of DNA, right? And they give the model of bDNA, right? However, this proposition was also based on the observation of Erwin Chargaff's law. He gives the equilibrium law, right? Equilibrium law is, he said that purine is always equal to pyrimidine, right? Or we can say purine upon pyrimidine is equal to 1, right? Ratio. The ratio between adenine and thymine and guanine and cytosine are constant and equals to 1. Are constant and equal to 1. We can say, hey, A upon G is equal to 1, or C upon A upon T, it is, right? Adenine and thymine. Or C upon G, it is also equal to 1. He gives this one, constant, always constant. Erwin Chergaff, Murik Wilson, right? And another is Rosalind Franklin. Rosalind Franklin and Lewis Green, you have to read these names. They gives us X-ray data structure. And with the help of this X-ray data structure, Watson and Francis Crick, they give model in 1953, right?